For decades, the consensus in textbook biology was that the adult human brain was a static organ, incapable of generating new neurons once development ended. However, recent observations suggest that this process remains active throughout our lives. This diagram illustrates the specific anatomical zones where this activity is concentrated. New neurons originate in two specialized niches: the subventricular zone, lining the lateral ventricles, and the subgranular zone of the hippocampus. Look at this cellular evolution pipeline. Stage 1 begins with proliferation of neural stem cells. In Stage 2, they differentiate into immature neuroblasts. Stage 3 drives maturation, developing dendrites and axons. Finally, in Stage 4, mature neurons lock into existing neural circuitry. To force this pipeline to operate at maximum capacity, specific biochemical catalysts are required. Proteins known as neurotrophic factors, specifically BDNF, NGF, and GDNF, act as the biological signaling molecules that command these neurons to grow, survive, and differentiate. This schematic details the required payload. Stack A targets neurogenesis support, utilizing compounds like lion's mane mushroom, curcumin, and magnesium L-threonate for synaptic plasticity. Stack B fuels stem cell generation, combining spirulina, astaxanthin, and blueberry extract to enhance cellular production. We know the exact compounds needed to trigger brain repair, but if you take these complex macromolecules orally, the digestive system's first-pass metabolism aggressively degrades them before they ever reach the bloodstream. The challenge of applied neurogenesis is no longer discovering the right compounds. It is strictly an engineering problem, bypassing the body's natural defenses to deliver intact payloads directly to the brain. To solve the payload delivery problem, we have to analyze the specific anatomical barriers that isolate the central nervous system from the rest of the body. The first barrier is external. The stratum corneum's dense lipid matrix actively repels standard supplements. Internally, the blood-brain barrier uses tight junctions and astrocytic end feet to block circulating systemic payloads from entering the central nervous system. To soften the external barrier, chemical permeation enhancers actively disrupt the skin's lipid matrix. Node 1 shows DMSO destabilizing the rigid bilayers to create openings, while Node 3 shows oleic acid fluidizing the matrix to facilitate unhindered drug diffusion. Chemical permeation works for small molecules, but the heavy, complex macromolecules required for neuroregeneration cannot slip through these chemically induced gaps. Delivering them requires active mechanical force. This advanced, multi-layered transdermal patch actively pushes large payloads through the skin. Structural microneedles create transient micropores. Above them, sonophoresis transducers emit ultrasound waves to increase tissue permeability. Finally, the iontophoresis engine's microelectrodes drive charged therapeutic molecules downward into the capillary beds. While each layer provides a specific advantage, they address different bottlenecks. Microneedles create the pathway, but rely on slow passive diffusion. Ultrasound increases permeability, yet lacks a directional force. By coordinating all three, the system actively drives the payload through the pores it just created. While this hardware successfully forces macromolecules into systemic circulation, it doesn't solve the blood-brain barrier. Accessing the brain directly requires bypassing the bloodstream entirely. Olfactory pathways provide a direct anatomical vector to the central nervous system. Using inhalers, the aerosolized payload bypasses the BBB completely. To survive the mucosa, payloads are protected by lipid-based nanoparticle encapsulation, while exosome vectors use biological tropism to target neural tissues. Bypassing the skin and respiratory tract entirely, engineered probiotics map the gut-brain axis to trigger neurogenesis remotely. Synthetic strains in a resilient microencapsulation shell survive stomach acid to colonize the gut, producing BDNF and SCFAs that travel up the vagus nerve. By deploying transdermal patches, respiratory aerosols, and engineered probiotics concurrently, we effectively surround the physiological barricades that have historically blocked neuropharmacology. This diagnostic matrix outlines the exact delivery logic. The selection of a transdermal, aerosol, or probiotic vector is strictly determined by the payload's molecular weight, the required onset time, and the specific physiological barrier being targeted. Proving the biology in a lab is only the initial hurdle. Deploying this to the public requires scaling up to good manufacturing practice, or GMP, compliance. Look at the left side of this manufacturing flowchart. Producing active patches requires layer-by-layer substrate assembly, integrating microelectrodes through photolithography. To ensure commercial viability, this complex integration must scale efficiently, with a peak production cost capped at $5 per unit. These transdermal and inhalable systems represent the immediate translational pathways. They are the deployable technologies engineered for commercialization right now. This timeline visualizes a 22nd century framework for extreme subcellular brain manipulation. We'll see CRISPR gene modulation alongside optogenetics controlling neural activity. The ultimate endpoints involve brain-embedded nanobots crossing the BBB and quantum neural interfaces manipulating neuronal patterns with ultra-high precision. Mastering these delivery vectors is the foundation for everything that follows. By bypassing the biological barricades of the skin and blood, it becomes possible to treat brain regeneration as a solvable engineering challenge.